Maria said, I'm going to give you a talk about how to teach, how to fuse art and science. In fact, I'm not going to do that. I'm a scientist. I'm trained as a molecular biologist and biochemist. I don't know any art. But then I was told to teach 3 courses for artists. So what I'm going to tell you tonight, share with you, is to share with you what I learned from art students and artists. Right? It's kind of it's a journey for me. It's kind of you know, when you reach the middle age, you just want to suddenly change everything, right? Change your field, and this is what happening to me, right? This sudden exposure to artists and art students has changed me profoundly, both to my personal life and also professionally. And I think what the impact of those people, artists and art students, art educators on me, can kind of symbolize maybe a bigger effect of artists on kind of the society today, right? Our society is really dominated by engineers and scientists, and we believe in progress and inventions and kind of, but then we kind of ignore the artistic side of things. What I learned in the past 4 years, I think other people in the society should also learn, right? So I'm going to share with you a few things that I have learned. Is that okay? Mm-hmm. So I'm a professor. I was told to always give 3 points at once and then summarize. So please don't mind if you can't see the slides. Tell me to shut up, and I'm rubbish. So basically that's it. I'm going to introduce a little bit about myself. By the way, who are you? Anyone who are biologists or scientists, or kind of
aspiring biologists.
Wow, great! We're all aspiring biologists. And any artist? Thank you. Welcome. Mariana, you artist. Any kind of educators? I guess, wonderful. So, nice to meet you. My name is Yunhua Lam. I'm originally from Hong Kong. I'm kind of a Hong Kong boy until the age of 20. I left Hong Kong and I went to Scotland, and I spent 10 years there. I love it there. I met many wonderful people, drink lots of whiskey. I drink several body weight equivalent of whiskey, and then I came back, and then I joined City University. I was trained as a biochemist, so this is my previous life. I give talks on, for example, how to use sophisticated mass spectrometers to sequence proteins and identifying genes that are differentially expressed in cancers, and purify bits of subcellular structures and analyze the biochemical composition, cloning microRNAs from tissues of different diseases, and look at what happens if you are infected with a virus and things like that. I give talks like this. I generate materials that will guide stem cell differentiation. I still do that. I'm kind of paid for doing that. But then, but I'm living a life as a kind of a scientist, a professional scientist and professor. But my problem is I love talking to people. So for most people, you're either a scientist or an artist, right? But then I like going to the pub and I kind of talk to people, right? So I believe I'm one of the few scientists in Hong Kong who are not embarrassed and shy and afraid of speaking to artists. So as a result, I collaborated with artists. So for example, there are 2 pieces of work that I collaborated with a dear friend of mine. In fact, I invited her tonight, but her name is Ching Wong. Very, very fantastic painter. Painting was her kind of forte. But she's very, very interested in science. So in fact, she's interested in reproduction, both biological and industrial. So she liked to push herself, her body, to limit. So all her work are about reproduction, right? So for example, she painted 100 bananas, 100 image of nude mice. For the biologists, you know that they're mice with no immune systems on petri dish. And every day she paint one of them. She won't paint more than one. So 100 paintings would take 100 days. And she follow particular ritual of eating particular food for that period. So basically, that's her work. And I believe every day we discuss about the biology and why the banana we're eating now cannot be produced, right? They're industrial product. All the banana we're eating are infertile. They're not normal. So these are interesting projects, right? So until one of the guy I met at CityU, basically, he's a dear friend of mine. Unfortunately, he's not in Hong Kong at the moment. His name is Hector Rodriguez, and he's a professor in the School of Creative Media. He's a digital artist. I don't know, some of them may know him or have been to his shows. I love him dearly, and he told me a story that kind of changed my life. I don't know. My life has been changed several times a day, so it's not a big deal, right? But then the following story changed let's say, opened my eyes. I would like to tell you the same story. Is that okay? In fact, I borrowed the slides from him. So so in rivers, in Europe, there are lots of flies, especially in the summer, right? Basically busting with life and flies, and there's called cantus flies. And of course, flies, they don't just born as flies, right? They start from embryo and follow several kind of steps in their life cycles. And one of the steps is called larvae step, right? And they are like a worm. You know, they look like that. They look really, really ugly. Very small, very small. I think the size is like 1 centimeter in length, and they live for briefly until they metamorphosize into a fly and fly away to freedom. So, but the interesting properties of this caddis fly is these larvae, they're very, very shy. So they other animals along the river, they like to eat them, right? Because they're just a piece of meat, right? It's a naked worm. So they don't like to be naked worms, right? So they like to gather stones in the riverbed and glue them together around the worm to build a tube, right? And this is interesting because these are the tube built by the fly larvae, and they're beautiful, right? You can see they're so regular, so they actually don't only use stones, so they use whatever they can see. So for example, if there's some shells or even snail shells from snails around, they will use the shell to build. So basically, they build the tube, the architects, they build the tubes with whatever material they have, right? So Hector told me, there's an artist from France, I'm not even pretend to pronounce his name, right? Let's say an artist from France. He did the following things, and when Hector showed me the video that I'm going to show you later, only a small clip, all the hair on my arm stood up. So I'm going to show you. So what he did is as follows. This worm will build a tube with whatever material they can lay their hands on. So what this artist was asking is, if I give them pieces of gold, can they do that, right? In the beginning, he used corns, but corns are too big, so they use pieces of gold leaves. So it turns out that they can do it. They build a tube with gold. And if they give them a pearl, what would they do? They incorporate these jewels into the tubes. So the end result is an art show. It is a kind of a jewel, a tube made of gold and diamonds and pearls, but it was designed. But the jewel designer is not any human designer, but the worm or the larvae. I love this work, and as a scientist, I immediately ask Hector lots of questions. For example, what's the chemical nature of the glue secreted by the larvae, right? I would like to have a chemical analysis on that. And also I said, why is the worm like to build, for example, this pearl around a particular region, right? So do they prefer pieces or kind of material of a particular size relative to their body length? So this is kind of a typical artist, scientist questions, right? Nobody knows, and I love to get my hands on larvae to do some chemical analysis. But if I told myself to think kind of outside my previous identity as a scientist and just look at these pieces of work, I see beauty. I see a collaboration between man and nature. I see how an artist, a creator, is supplying material for an organism and ask the organisms to build things that are completely out of expectation. The result is beautiful, mysterious, surprising, and just wonderful. So I told Hector the following things over a cup of coffee because he doesn't drink, and he said, "Well, Yun, if you like that, I would like to invite you to my school." So that's the building, right? That's a wonderful building. It's part of our university in Kowloon Tong. If you kind of are over in that area, you're welcome to visit, right? It's a beautiful building. So, and then Hector told me, well, just by coincidence, because you're interested in this artwork, just by coincidence, the School of Creative Media, which is a kind of the art college of our university, I've never been to that building before because it's far away, and also I'm an artist, right? I can't be seen together with those guys, right? But then Hector said, if you like that project, in fact, the school was at that point building a new program, and this program is a revolutionary program. It's the best. It's the only one, definitely the only one in Asia. We're trying to see whether others. I think we have done some investigation. I think there are. Anyway, it's a program called the Bachelor of Art and Science, bracket New Media. So in other universities, there are MA program, Master of Art program, that fused between art and science, especially digital art, so it's nothing new. But what they, at that time, were proposing to me is something revolutionary. Number one, they already have a science program, so to speak, about computation, about computational programming, mainly training students to become a digital artist, animator, and video game designer. So they're already very, very strong at that point. They have already a degree in that area. But what they're trying to do is to teach students new media, meaning wet lab, so-called laboratory-based science. In a way, DIY science. So what they want to do is to gather some students, art students, who want to become an artist. But these students, they don't want to become a painter, a sculptor, or kind of artist using so-called traditional medium of creation. They want to use science as the medium of creation. They want to build art from biology, from physics, mathematics, and other disciplines of sciences. That is kind of quite exciting, isn't it? So in fact, if you look at the website, you can see that this major is especially designed for students who are not afraid to break down the barriers between art and sciences, right? in fact, this statement is also, I suspect, the reason why we're here, right? We're here because we want to break some barriers. We want to do something that are outside of the box, and we want to see in the gaps between kind of traditional breakdowns of disciplines and compartmentalization of knowledge, can we do something interesting and never be done before? So, to cut a long story short, after that meeting and visit, my life has changed, right? So, in fact, I now know that because nobody wanted to teach that, so I was kind of asked, told to do that. So we co-developed the curriculum together, and now we have 3 courses, or I contributed to 3 courses. So the first one is called Introduction of Science to Artists. The second one is called Artists in a Lab. I love that title. And the last one is Biology and Art, the one that Marian helped or contributed. And they have these names, the university courses, so they're these code names. So I'm going to tell you something about these 3 courses, but I'm not going to pretend to you that we have great courses and everybody's happy and wonderful. So this program was established 4 years ago. In fact, I participated in this program for 4 years already. The first year we did that, I have nervous breakdowns 3 times a week on average, because we get some students and we simply lie to them, told them it's very interesting and exciting, with good career prospect. So this student camp, they chose they chose to major in our project. But then, to be honest, we have no idea what to teach them. We have no idea what to teach them. Imagine you're teaching engineering. So, university have been teaching engineering for 200 years, right? Let's say if you're teaching biology, I'm a biologist, and I can probably sit down and kind of write up a curriculum for a biology program, major program in half an hour. Not because I'm particularly good, but because we have all the university are teaching biology using a similar curriculum anyway, right? From molecules to organisms, from genes to environment, from simple to complex, so it's easy. But if you want to teach a course about art and science, if you Google it, nobody have done that before, right? We are the first, and well, it's kind of good to say that we are the first, but then because nobody have done that before, everything we're teaching these people was taught for the first time. So I still remember the first class. I did the first class, right? And I was telling them, biology is very, very important, and chemistry of life, and we are living organisms, but in fact our cells and bodies are made up of chemicals and chemical reactions. And I show a kind of simple molecules, right? It's basic organic chemistry. I don't know anyone who has studied organic chemistry before. Carbon linked to carbon, right? And there are different ways of linking 2 carbons together, and one way is carbon by double bond, right? So that double bond, and the shorthand is a chemical formula: C double bond, right? To me, I don't even remember when I learned it, right? It's kind of a natural, the most basic alphabet for chemistry, and to explain life you have to explain chemistry, and you have to learn the basic alphabet, right? So I thought I gave a reasonably good elementary lecture on organic chemistry until some sleeping, and then afterwards one student came up to me and said, that slide you show, carbon, double bond carbon, what does that mean? So if carbon equals carbon, What does it mean? And is it an IQ test? Because the two carbon is exactly the same, and there's an equal. So why are you telling me that C equals to C? So of course, basically, he never heard of carbon double bond before, right? So to explain to him, you have to explain chemical bond. You have to explain to them the different kinds of chemical bonds. But then when you do that, you can see that he started to look away. He started to look very, very tired. And then he started to look at his watch. And then in the end, he said, "Oh, Dr. Lam is very, very nice, but I kind of need to go." Right? Completely not interested. So that's how we passed our first year. Very, very low kind of student satisfaction rate. We are crying and we are not getting anywhere. So the second year we start again. We completely changed our way. Instead of teaching them carbon double bond, carbon, or any other fundamental in chemistry and biology, we scrap them all. Come on, they're not going to be scientists anyway. They're not going to become a doctor, engineer. They're an artist, right? Why should they know so much biology? All they need actually is to know enough biology or have enough curiosity to be able to enable them to collaborate with a biologist and do some collaboration together. So the first realization is not to turn them into scientists, but turn them into a person who's open-minded enough to speak to scientists, just like I was speaking to Hector in a pub, right? So that's the learning outcome, right? How to speak to a scientist in a pub. So, and then the more we do that, the more we feel surprised, sometimes really surprised, sometimes happily surprised. So for example, here is an example of the surprise element. So we're teaching them, I don't know any biologists in the audience. So this a protein gel, right? It's kind of a gel electrophoresis. So you make a jelly, and then you kind of use this jelly and so-called an electric field to separate macromolecules, right? DNA and proteins. But then DNA and proteins have no colors, right? So after the separation, you take the gel out and soak the gel into dyes. Some dyes will only reveal the DNA. Some dyes will only reveal the proteins. And these blue dyes reveal the proteins because the many carbon double bonds, it doesn't matter, because the dye will bind to only protein specifically, right? So we're going to teach them this, but then everybody's sleeping and jelly, no interest. But then some students, after learning this, came to me and said, well, we have an idea. And the idea is as follows, right? I was very, very surprised, but I was crying actually when they told me, right? Laughing and crying. So the idea is they want to watch some sad movies, right? So no more, first of all, no more gels, right? So they want to watch some sad movies and cry. So top 10 sad movies. Do you know what's number one?
Titanic?
Huh?
Titanic?
No, it's not even close. The number one movie I never watched myself is called 7 Pounds. Is it sad? This one is 11 Let You Go.
Oh, yes.
This one is number 2. Anyway, they watch all 10 of them, right? They find 4 friends, anonymous friends, to watch all 10 movies, and then cry into. So, and then they took the tissue. So after watching, they kind of collect the tissue papers, and then, well, because they're scientists, right? So very, very precise. So they put the tissues by using a forceps into a bag, right? So basically, it's an exercise where they have to write up a scientific experiment, right? How to learn how to correctly report some technical details. So then they put it into a fridge, right? And then they bring it to the lab, and then they use the dye that was specifically labeled proteins to stain the tissue papers. So what they wanted to do is, they're not interested in your protein gels and bands, macromolecules that we as a biochemist love. they want to stain the tears on the tissue papers of these 4 students, anonymous students. I suspect it's themselves, but so basically, it is an unexpected and playful use of a standard scientific procedure, right? I'm a scientist. I never heard of using that. Of course, there's no application, but to them, it's a very important jump, a leap in their thinking. They're using what they've learned in this class, a dye that specifically label proteins, but then what they're labeling is not proteins in the gel. What they're labeling is a record of their emotions when they're watching these movies. To them, as science students, this is more important than cloning genes and curing cancer. They want to find a way that can record the tears of their when they watch the movie. Do you want to see the tissue? This one cry a lot, but this one kind of like still like you can actually see kind of is almost the same shape, right? So basically, maybe a teardrop in the eye, like tapping, and this one is a flood of tears. It's interesting, right? In fact, we like it. We like how it turns out, and we love this kind of unexpected and playful deviation of a standard procedure. So what I'm going to do in the rest of my talk is to show you more students' projects. Is it okay? Some of the student projects are, to me, laughably crazy. They're all crazy. Some of them are laughably stupid. Some of them, to me, is quite touching. So is it okay? Bear with me. About 10 projects. In fact, there are actually lots of projects. So here, I forgot to tell you, once again, I'm completely disorganized. Instead of teaching them carbon, double bond carbon, or basic things, we put them into a lab and teach them a number of labs, basic things, in a way, DIY biology. We teach them how to isolate DNA. We teach them how to grow microorganisms, including bacteria and fungus. We teach them how to look at cells under the microscopes and various things. So so basically they do that. It's very easy, right? If you have done DIY biology, you know, you follow the procedure, you get somewhere. But what we told them is, after you have done this lab, you need to create an artwork that is based on this lab or that utilize the technique you learn in that lab, right? So no background, no boring lectures, but just do the lab and then do the creation, and then tell us what happened. So there's one example. The students need to choose one of these things, right? So from patterns and variation in biology, information is beautiful, to I love this one, time. So basically, the students have to choose one of these 9 things and then do art project. And another condition is they have to based on a lab technique they have learned in the course. So I don't have time to tell you all this, right? So I'm going to just choose one theme. Guess which one I'm going to choose?
Area A.
Area A. Great, because it's my area. Area A, because I love that, you remember this flies and larvae and making the goat. I love it so much. In a way, that's collaboration with other organisms, right? So I'm going to tell you 10 projects very quickly of how students develop their artwork using this theme. Is that okay? So the first one is about bacteria and fungus. The student learn how to grow bacteria and fungus. They simply collect doing a swab. I think you're probably doing that also in your workshop, right? Very simple, using a sterile kind of a mop to collect microorganisms on surface, and then we plate on a petri dish and to grow, basically. It's a 3-hour student lab. And the student like it, at least I hope. So I'm going to tell you a few kind of development. By the way, none of the projects I'm going to tell you, the idea came from me or any other teaching staff. All the idea came from the students. So, and this is a very, very fundamental difference from traditional science course. And I'm going to tell you a little bit why I think this is important in the end of the lecture. So the first project is easy. In the beginning, in our course, we collect microorganisms and put them on a petri dish and count the number of colonies. And the number of colonies will tell you how dirty that surface is, right? So this student, she's asked obvious question that I'm sure you also asked, right? She's a painter, and she said, well, can we use the bacteria as ink and to paint, right? So it's kind of boring, right? So, so basically, by the way, all the snapshots, all the slides from now on were made by the students. I just copy from their student report. So basically, they learn how to grow the bacteria, and then she simply learned how to grow the bacteria in a liquid culture, and then use the liquid as ink. So this what she made. It's beautiful, isn't it? It is. To me, beautiful, and at the same time, the word changed over time, right? As the colony grow, the word will become more and more obscure and disappear. So, so basically, this is one project, right? So it's kind of a interesting first step, right? It's actually the simplest project tonight. So the second one, by the way, these are all the name of the students, right? So the second one is another kind of development from the simple bacteria plating, and that one is also something that you always want to play with when you are small, right? You can mop a surface, right? And then you get the bacteria from your mop, and then plate on the petri dish and count the number of colonies. But then you can also do something like that, right? I'm sure people, we all know that our skin, our body is full of bacteria, right? And the bacteria are good for us actually. So, and in fact, instead of mopping our skin, you can simply touch our skin on a petri dish, and then you can see basically this, and then grow the petri dish for a few days, and then this you can actually see the bacteria colonies from our skin, right? The interesting, so she's fascinated by these kind of things. It's a childish variation, modification of our procedure, but she's fascinated because she said that bacteria, for example, that is not one bacteria, there's not one bacteria, but is a colony of bacteria that came from a single bacterium, but that originally came from her arm, right? But the position of this bacterium in her arm, here, is a replica of the position of that bacteria in that position of her arm, right? It's almost like a photocopy of the spatial distribution of bacteria on her arm onto the petri dish. I don't know what's so funny about it, but she's fascinated. So that's a student, and she said, oh, I don't want to do my arm, I want to do my face, right? So in fact, this is actually another step, one step ahead, because her face is much bigger than the petri dish, right? So she can't just do that. So what she did is she used a cloth, which is a disinfectant cloth, to put on her face first, right, to copy or transfer the bacteria from her face to the cloth with a spatial kind of fidelity, and then put the cloth on the petri dish, right? So she used the cloth as a kind of a middleman, right? So this is a one-to-one replication of the spatial location of the bacterium from her face to the petri dish. But it's not the end, because she said this is not her face, right? What she did was copying the material from her face, which is a 3D surface, onto a petri dish, which is a flat surface. So she doesn't like it. So then she said, why must a petri dish be flat, right? A petri dish can be three-dimensional. So this is what she did, right? So she decided to make a mold of her hand, and so basically using a pasta, basically it's like primary school art lesson, right? To make a one-to-one negative replica of her hand, and then to use this cast and pour agar to make a one-to-one replica of her hand, right? So this is her hand. So we have the replica in the lab, and she didn't know at that time it's also replicated her fingerprints. So very, very accurate. I'm going to show you, hopefully it's not too dark, right? And because she's an artist, so she actually designed a light box to show her hand. So now, after the hand, so this is her hand, one-to-one replica, it's actually quite spooky because it just looks like her hand. So now the next step is to use the cloth technique, right? So she buried the cloth, actually is a glove, right? So basically transferring the material on her hand to the glove, and then turn it, and then use the hand to wear the glove, then is an accurate one-to-one, spatially accurate copy of the material from a hand to this hand, right? And then afterwards, we incubated this hand to see what happened. So this is her hand. Unfortunately, it's too dark for you to see, right? And you can see it's a time-lapse movie, so you can see the material coming up. These cornelis are from single bacterium that are in the same position of a head. Yes?
Were they incubated?
Huh?
Were they incubated?
We incubated in a oven, actually, 37 degree. Basically, there's nothing, it's no different from the oven at your home. There's 37 degree, but of course we put a camera in there, right, with lighting as well. Once again, for an artist, it's interesting because it's kind of an out-of-body experience, right? It's kind of mapping the material distribution. But then for a scientist, we also think that it's kind of an interesting project. Nobody have done that before. So now, for the first time, we can, for example, ask whether you guys are doing DNA barcoding, right? We can ask for the first time whether the bacteria on her 10 fingers, are they actually the same species or same mixture of species? Or even the bacteria from that part of her hand and that part of the hand is kind of dirty, right, between the gaps of the finger, and that part of the hand we wash all together. Are they actually evolving different types of bacteria? So using this technique, we can select cornelis from different positions of the hand and do DNA barcoding and see what happened. So this student, she really loved this project, and she's going to do version 2 of this project, right? It's the most ambitious project that we have done in our lab. So here is a prototype on her hand, but now, coming year, she's going to do her whole body.
Wow.
That will include doing the cast molding of her whole body, and then making the gel, and then cutting the gel into different bits, and then doing the glove technique, and then incubate independently. Afterwards, we put them back together. It's interesting, isn't it? Right? There's no application, but then it's kind of mysteriously beautiful. So we're coming up one level of kind of complexity. So another student, and she's not into body image and body art. Her forte is photography. She's a photographer. So she she liked to do Polaroid. I don't know whether, so I thought they discontinued this, but they obviously they're still making it, right? A Polaroid camera. For the people of Maria's age, you never heard of, right? Because you have iPhone, but it's a film technique, where it's a film-based photography, but then it gives you an instant development. You kind of develop the image from taking the image to having a print in a few minutes, right? Because the chemistry of film development is actually embedded in the film, right? So Amber, the student, bought this cartridge of films. I don't know, for the people of my age, we all play with that, right? And she learned how to open up the cartridge in a dark room, take out the film, and we told him on the surface of the undeveloped film, there's actually a layer of protein called gelatin, right? So, and we told them bacteria like to eat gelatin. So she learned how to do that, right? Basically, open up the cartridge of Polaroid film in a dark room. In the modern language, we call her hacking the Polaroid, right? But then I actually don't know what that means. At the same time, she also learned how to isolate bacteria, right? This time using traditional kind of cotton swab. This one is called Inside Me. I don't want to think what that is, but this one is called Eyes. So basically, she collected bacteria from bodies and from the environment and learned how to culture them. And then she cultured the bacteria on the gelatin layers on the surface of the opened Polaroid film, because the film has to be undeveloped for reasons that I'm going to tell you. So she have to open that and do the culturing in the dark room, and afterwards she have to put it in a box, a light-proof box that she made by herself. In modern language, she hacked the box, right? No, she made the box, and then take it out of the dark room because it's light-proof, and incubate the box in that oven for a few days, and afterwards she put the box unopened back to the dark room and reassembled the film and put it back into the camera. So why is she doing that? And the reason is, she said she wants to make an image of a person, but she wants the biology of that person to participate in the image creation process. So what she did is she make a series of portraits of her friends and also one portrait of a plant. And before she made the portrait, a few days before, she asked that person to donate some bacteria and culture the bacteria from that person, and then culture the bacteria on the film that she's going to use to shoot that person. And then take the image and then develop the image. Right? Do you want to see what the images look like? So unfortunately, probably the room is probably too bright. The images are very, very dark. So of course, it's not perfect, right? The images are broken and in a way bad, but in a very, very touching, almost moving ways. That's because the light box, she opened the film in the dark room, placed bacteria on it, and put it in the box. But the box is never tight, right? She made the box by herself and wrap it in the newspaper. So basically, the images are very, very overexposed because of the film is partly exposed. So it's actually the image of herself, self-portrait. She said, I shot this one 3 hours after I applied bacteria from my mouth on the left side of the film. Unfortunately, but the left upper corner is lost. So this is a record of herself together with her bacteria, right? The bacteria grow on the gelatin surface of the film and partly obscure the film. So, and this one, plant bacteria on its root. So you probably can see the bacteria on its root. this is kind of her work, right? She tried to make a image by both the subject and the bacteria around him or her. So that's in a way collaboration with other organisms. So next up, slime mold. Unfortunately, we're not ready to give you the slime mold, but we're very happy that you guys can look after it. Anybody have heard about these organisms? Yes. I think we are still the only lab in Hong Kong using slime mold. So slime mold is not a mold. Slime mold is actually an organism that grow in nature, probably surface of rocks. When there are lots of rains and kind of moisture on a rock, this organism will grow. It's not bacteria, it's not fungus, it's not a mold, but it's like a amoeba-like of organisms. So, and of course, they are single-cell organisms, just like bacteria or fungus. But when they grow, when there's so many of them, they will stick to each other like this, like cheese actually. And this kind of cheese-like material is a very, very interesting biological entity, because it's somewhere between. It's a very, very primitive in evolution. There's somewhere between single-cell organisms on the way towards multicellular organisms. They can't make up their mind whether they're single-cell or multicellular. But the interesting about slime mold and the fact that it captures imagination of many artists is of these properties. If you put the slime mold in the middle, this kind of cheese material will send out branches to look for the food. So it's almost like behaving like an organism. It will shoot out offshoot and digest the food, and this nutrition after digestion will be pumped back to the main body, right? Until all the nutrition is digested, then they will shoot out another one to eat another bit, and then shoot out another one to eat another bit, as if it's thinking. But of course, there is no brain. It's got no brain, right? So, an interesting thing is, if they want to eat something and kind of send out this kind of the offshoot of, if you put the barrier blocking this way, this offshoot will actually turn around and look for the food. If you put multiple barrier, they will actually turn around as if it's solving a maze. But then, of course, it's no brain. It's got no neurons and no brain. So a lot of scientists are kind of trying to understand how this kind of single-cell organism on the way to multicellularity are actually doing that. But the artists are fascinated because it's kind of a much more interesting than bacteria, because it can find a path, or it almost like it's got intelligence. If you haven't heard of this material before, please search for this term in YouTube. There are many, many interesting experiments by artists, and there's also a very, very famous TED talk by an artist who used slime mold as her kind of medium of creation. So, and of course, in the hands of our students, they are not, of course, they're not interested in how the evolutionary importance and how they solve puzzles. They're interested in politics. They're interested in mainly politics, as if this person. So, you know, slime mold can find a path, right? If they give you some food, if they have the cheese-like main body, and if there are obstacles between the main body and the food, the offshoot will kind of turn around and look for the food, right? As if it's finding a path in a maze. In 2015, there are also people who are doing the same, right? This student, her name is Bobo, and she was highly affected by the Occupy Central movement. For the people who are not from Hong Kong or who are not living on this planet in 2015, you probably don't know that there are two groups of people, right? Yellow or blue, right? If you're yellow, you are occupying the central. If you're blue, you are supporting the policeman beating up the yellow people, right? So, and for a scientist, when we look at slime mold, we're thinking about billions of evolution and the position of these strange organisms in the evolutionary history, billions of history. But to an artist, all she saw, she wasn't interested in all that. All she saw was the yellow color. Yellow occupies central. So she did the following project, and basically what she did is she said it would be wonderful to see if we make a map of Central, right, using Google Map, right, and use the streets of the map as a maze, and we put the slime mold in places where the people were occupying, and we put the food in the home of CY Leung, then what will happen? So she did exactly that, and we are kind of quite touched by how motivated she is. She just wants to see how the yellow slime mold is kind of rushing to CY Leung's home and eat all his food. So this, she just wants to see that, right? She spent months and months doing that. So this is actually from her work. So she used Google Map, and she used kind of Photoshop to map out the streets, and she used laser cut it's also maker technology used by a lot of makers to cut the street out, and then she basically built the maze, which is a scale model of the street of Central and Admiralty. Then she did her map of Central, and then she put the slime mold where the people are occupying. And then she did the time lapse. At that time, once again, if Maria, you want to do some slime mold workshop, growing slime mold is very, very easy, but what we find out is the time lapse that's difficult. Because I'm going to show you some tricks, mainly because of lighting. So, do you want to see the Occupy Central attack of CY Leung? So, I think this is his home. I think, and this is the people. I think these three spots represents the people occupied in the three main area of occupation. So basically, these are the people. Sorry?
How many days does it take?
I think it's one image in every 10 minutes for one day, something like that. They grow very, very fast, very fast. Overnight, after one day and one night, 24 hours, all the street is covered by yellow. It's actually a much longer movie, but then I think she did it at home and in her toilet, and her mother went to the toilet and switched off the light after her. So towards the end of the movie, suddenly it's dark. So, but anyway, that's her work, right? Once again, no application at all, but just have fun. So one more step of sophistication of complexity. So this student, and she was learning in other part of the lecture of a phenomenon called Schrödinger cats. You all know that, so there's no need to explain. Basically, if you close a system, you have no idea whether the cat is still alive or dead. It's a famous thought experiment on quantum mechanics, and the cat is either alive or dead, right? It's kind of an example of an illustration of a quantum state. So you also know that slime mold can be killed by blue light, which is strong light, UV light and blue light. But then if you use red light, they're fine. So she designed a project called Schrodinger's slime mold. So in which the viewers are not allowed to look at the slime mold, while the camera inside will look at the slime mold, and the viewer will push a button, and the computer will randomly assign the LED whether to give it blue or red light. When you push the button, you have no idea whether you are promoting the growth of the slime mold. Red light will increase the temperature, and therefore the slime mold will be more happy. But blue light, because it will damage the DNA, will kill it. The viewer, when they push the button, have no idea whether you're helping it or killing it until a few days later. So this is another example, I think more sophisticated than occupying C.Y. Leung's home, because she incorporates other part of her lecture. So the next one is collaborating with other organisms, next level. So basically bacteria, very kind of primitive organism, slime mold one step higher in evolution, then fish, right? It's a vertebrate, it's got a brain, 2 eyes. The next step is to collaborate with fish. And we teach them how to grow fish. These are zebra fish, which is a kind of a very nice model organisms for biologists, because we love to do genetics with them. We love to model, make disease model of zebra fish. But for the artists, they don't care about disease models, right? They don't care about curing cancers. They are interested more about how the fish responds to light and visions, just like how human beings are responding to lights and patterns and basically visual effects. We teach them something interesting. So for example, this is not by the students, right? It's from my teachings, it's from my slide. There's a phenomenon called optomotor response. So this is a zebra fish. They're made to look at these things, right? They're turning, and you can see that their eyes are responding to the movement, right? Because it's almost like showing a fish an optical illusion. Just like human beings looking at optical illusions, the fish are full and their eyes are following the pattern. So once more, so this is another version. So if you show the fish moving stripes, the fish will think the moving stripe is a river, is a water current, right? The fish are living in river, shallow river, and the sun will cast, the sun above the water surface will cast shadows in the bed. So basically, the water currents will be sensed, can be seen by the fish as patterns moving underneath it. So this is actually not from me, it's a Nature paper, it's scientific investigation of how the brain of the zebra fish works in response to this illusion. So we teach this lecture, and then we show the student how to do it. So, but how to do it, right? So once again, in modern term, we're hacking the systems. So we're putting the fish on a petri dish with a little bit of water. The fish are very, very small. And then we use an iPhone to show the pattern, right? So we make the pattern by some simple software, and we put this pattern, moving stripe, on YouTube. So the student will open YouTube, and then they will use their phone as the kind of as the basis and put the dish on it. And now the students are responding to the moving stripe as if they were responding on the current, kind of the patterns of current on the riverbed, right? So they've done that, and so these 4 students, they said, now that's interesting, because whenever the fish saw the black stripe moving below his eyes, whenever the back stripe, the band passed his eyes, his tail will stitch a little bit, right? So in that particular video, the fish were immobilized, so they are kind of pinned on the dish, right? Those evil scientists. But then the student realized that if you don't pin the fish down and immobilize it, when it stitch the tail, it will swim, right? Therefore, you can control the swimming direction of the fish by using the stripe, right? So now they realize they can do that, and this is what they've done, right? Scientifically, as a scientist, I don't think people have done what they have done before, but I'm going to just show you what they've done. So this is a stripe, this is an iPhone, and above the iPhone screen they put a petri dish with a fish, right? So what you're going to see, but by the way, there's a flashy image in the following movies, right? If you don't like flashy image, then don't look it. But just follow this fish. So the fish will move along the direction of the stripe, right? By the time it reaches this opening, they change the direction, the fish will move along. They change the direction, the fish will move along. And then there's one funny bit, they make a mistake. So now, maybe the fish is a little bit tired, but now they make a mistake, they change too fast. No, doesn't matter. The brain of zebrafish have about 4,000 neurons. This is actually much simpler than human brain, right? Only 4,000 cells, and scientists know pretty much how all the cells connected to each other. In fact, scientists knows when the fish are exposed to this moving stripe pattern, how their brains works, and which neurons are firing signals, and how the eyes receive signals, put it in the brain, and how the brain processes the signal and control the movement. Now, in a way, in modern terms, we have hacked the brain of the zebrafish. So, in a way, this student is able to control the movement of the zebrafish as if it is a robot. Every time it works, right? It's simply by hacking the brain of an organism. So, that's interesting, right? So now, how about upping the game, right? So what's next? So there's another group of students, 2 students, and now instead of putting the dish on the screen of an iPhone, how about surrounding the fish vertically with the illusion? So this is how it works. Unfortunately, once again, it's maybe too dark. So you can see the fish gradually rotate along with the image, right? So, of course, for the people who are interested in prehistoric cinema, you know that this is actually not invented by them, right? It is called zoetrope. So basically, it's like that. It's cinema before the invention of cinema. So it looks like this, it works like this. But if you look in the gap, you can actually see illusion of moving. So what these students are doing is expose the fish to optical illusion and see whether the fish, if a human viewer is looking through the gap, how the fish participate in the creation of this illusion. It's double illusion. It's illusion for the fish and illusion for the human viewers, right? So it's kind of optical illusion. So how to up the game? So next one up. The next one up, in a way, is scientifically, in terms of design, much simpler than the previous one. But I personally love it. I love it. While other people are using the stripe and try to control the fish, this student, his name is Nero, and I love him, man. He asked the following question: why must we control the fish? Why must we control the fish? Right? So we hack the brain. So what's so good about it? We can control the fish, where to swim, where to stay, where to turn. What's so great about it? we are synced into, once again, the control freak habits of human beings, kind of casting our power over other organisms and make fun of it. What he wanted to do, to me, is technically a low level, but spiritually much more sophisticated in all the other work. What he wants to do is to ask the fish simply to show the fish beautiful images he created in response to a piece of music, and ask the fish to swim over it. So it's just a clip. It's on YouTube. It's just a clip of what he has done. He wrote a program that converts the music into this pattern. And he simply wants to study and appreciate how the fish respond to this environment. So the original is much longer, and also there are several songs. I love this work. It's one of my yes.
Did you try with different songs?
Sorry.
Did you try with different songs?
Yes. He also tried different angles and also different types of music.
Did he discover what kind of frequency that the fish actually like?
I asked him the question, and I asked him the same question, and for science students, they would say, oh yes, I'm going to do that, and systematically investigate the effect of music frequency. And then I asked him that, and he said, like that, he's smoker. And he said, I just don't want to do that. He said, the fish are fine, right? So in a way, to me, this is a real art and science project, right? He's not controlling everything, but he's trying to create an artwork or a world where humans and fish collaborate and complement with each other. So next one up of collaborating with other organisms, and that organism is human, is the last example. So she's a student who did a very interesting project, right? And that project is about measuring the size of pupil of your eyes, right? By using a high-speed camera. So you can see the pupil is here. So basically, she, as an interviewer, will ask 4 friends independently a question. So it takes them, and he asks the question without knowing the answer until 10 seconds later answer the question. So she knows the answer, but she recorded the first 5 seconds after the question, right? So when the interviewee was thinking about the answer, she's imaging his or her eyes, right? And she's using a software, ImageJ, also a friend of DIY biologist, to measure the change of the pupil. So this is the pattern. This is the answer, pupil size, and this 250 images over 5 seconds, right? She asked 2 questions. One question is represented by green bar, and one question is represented by blue bar. Do you want to know what the question is? What's the question?
What's the control question?
The control, she never told us. So she said the control question is not related to love. She need the control question because she need something to compare with, right? She showed out of 4 person is kind of hardly a kind of a statistically significant answer. Every time when you ask, are you in love, the pupil dilated more than a control question. The interesting thing is, the dilation, this trend, was also measured no matter the answer was yes or no. when you think about love, either happily or kind of with despair, your body responded in the same way. So in a way, it's almost like a scientific investigation, right? So the next one, unfortunately, it looks too spooky now, but if you have a darkened room, it's much better. She's a student called Grace, and she learned in one of the lectures that artists associate color with emotions. For example, red is associated with anger, and love is associated with magenta. It's not by scientific investigation after interviewing thousands of people, they have this association, right? So she learned that in the lecture. So what she did is very interesting, a little bit complicated. I would like to explain. Grace is a person who loves writing. So she's an artist, but writing and words is her first love. So one of her favorite books is a book written by a post-war Japanese writer called Setting Sun. I don't know what anybody have read it. He's a very famous writer. He died before his 40th birthday, and this book was published after his death. In fact, he died by killing himself after 5th attempt. So anyway, it's a very sad book, right? But it kind of also very interesting book in terms of literature, because it's a novel written in the form of letters and things like that. But anyway, Grace loved this book, and what she did is she copied every sentence from that book, and so when she was reading the book, right, and there are tens of thousands of sentences, so whenever she read one sentence, she recorded whether she's angry, surprised, happy, fear, sad, or laughing, right? So basically, she recorded her emotion when she was reading this book, sentence by sentence. And she copied the sentence, and she coded each sentence according to her emotions in that particular reading. So it's okay so far? So then she go around with her computer. She uses the camera on her computer, because she doesn't have a mobile phone. So, and she make video, right? So now she have a video, and she also have these sentences, and also this assignment of emotion for each sentence, right? You get it so far? So now she will play the movie, and she wrote a program to measure the color intensity of different color in each frame, every second of this video. And afterwards, she calculated, because of this color emotion assignment, whether the emotion is surprise, or I think later is honesty, anger, right? So basically, there's a calculation every sentence, every second. And each time there's a calculation for a particular emotion, she will randomly select amongst her sentences of that particular emotion and put it there. In a way, when she's showing the video, it's a poem coming up, but she has no control of the words in that poem. All the sentences of the poem came from that book, but the order of the sentences were dictated by the video and the color intensity, the intensity of different colors of the video. And because there's so many sentences for each emotion, and these sentences were selected in random, so she has no control over the exact content of the poem. Every time she showed the same video, a different poem will come up. So this actually her work, and in a way, it's a very kind of different reading of a book, right? She read the book with her life, with her, she used the book, her favorite book, to illustrate what she sees when she is taking a walk in town. That's interesting, isn't it, right? Not bad for a first-year student. Collaboration with herself, and also collaboration with a writer who died 60 years ago. So, is it okay? Right? I think I talked for too long. I'm going to tell you one quick bit before the end. Is it okay? We will take 5 minutes. Why do I think what I learned from this artist? Don't you think these art students are great, right? It just surprised me. If I supervise science students, even PhD students, I have to tell them what to do. I tell them what to do, and they will do a perfect job because they are very, very good at following protocols. But they never come up with an idea. But the art students, they don't know how to do anything, but they always came up with an idea, right? So this is a special map written by some psychologists, education psychologists from the from Berkeley, UC Berkeley, and to explain to STEM educator what is scientific investigation. So according to this educational psychologist, there are three. Basically, STEM is about testing ideas, right? You have a practical problem, you have kind of exploration and discovery, and then you test the idea, feedback, and there are also two things. One is called benefits and outcome, whether your work is useful to the society. And then community analysis and feedback, whether your peers think your paper can be published, right? So feedback, and then this cycle will turn, and then you create new discoveries, new inventions, and new ideas, and progress in mankind. Well, and then if you think about, and we look at what do we mean by exploration, and what do we mean by testing ideas, they said that testing idea come up is about gathering data, interpreting data, right? Hypothesis, expected results, actual results. Scientists are really, really well trained in that part. We are really, really good at investigating something. Just tell me what to do and I will investigate. Just give me a question and I will do a good job. But the problem is we relied on other people to give me the question. Either the other people are doctors who said there's a difficult cancer they cannot cure, or environmentalists who said there's some kind of pollutions and they don't know how to clean it up. The questions are always from the outside. We are very, very good at executing the experiment, but we don't know what kind of questions we should answer. So, also, scientists are always pulled back by peer analysis and benefits analysis. We are always pulled back by how other scientists think we should not do it or we should do it. We care too much about whether what we do have no application or actually there are applications. Therefore, scientific investigation in the 21st century has changed from the sheer joy of making discovery to an investment. The community invests money to my lab, but they are wanting some results back. If we can't pay back those investment, we're in trouble. However, artists is after 9 o'clock, so I can swear, right? Artists doesn't care about shit. Artists care about making interesting experiment. Artists are interested in playing with the medium. They're interested in making things that they can play with. In fact, this kind of ability is exactly what we need. So according to the Berkeley psychologists, exploration and discovery is about making observations, asking questions, is about surprising observation and pure chance, is about practical problems and curiosity. Science education in the 21st century are extremely poor in teaching people how to become curious. But artists They're born to be curious. So therefore, I thought I'm going to teach artists science, but in a way, I learned so much from them. Because they teach me how to play with things. They teach me how to forget about benefits analysis and peer pressure. They teach me how to have joy again when you're seeing things that you have never seen before. In the end, I'm going to show you one video. The video was made by a student. I've shown this video many times, so for the people who've seen it, please forgive me. This video tell me what is important and what is not important. But before I show you the video, I need to explain a little bit. I teach biology and I think I know everything. And I think I know, wow, the DNA sequence and genomes. And one day, after teaching a class in basic plant physiology, how the plant get the sun and then get energy and get water from the soil, everything is solved. One student came up to me and she said, so I learned about plant science, it's very good, but Dr. Lam, have you ever seen a plant that will wave at you when you talk to it? Have you seen a plant that will wave at you when you talk to it? I said, oh, come on, go away. This kind of, you are crazy, right? What have you taken this morning? But then she said, well, no, because I've seen a plant, when you talk to it, it will wave at you, right? So, movable plant, for example, plant that bite insects, I don't know whether it's fly traps, has been documented, but there's kind of different type of waving at you when you talk to it, right? Because it involves some kind of perception of sound and understanding of what you're saying and transform your greetings into action, right? It's not what a plant can do. So I said, go away. And then the next week, she showed me on her mobile phone the following video, right? I'm going to show you this. So look at these two leaves, and also look at the smaller leaves around it, right? Notice that all the other leaves are not moving, right? It's just this particular plant. So that's the end of the talk. I will tell you what is important and what is not important. For the people who can speak Chinese, you can probably see that they're gossiping about the plant, right? For the people who doesn't speak Chinese, I think you can still appreciate the joy in their sound, right? In their voices. They show something that nobody have seen before, and then they're the first one who discovered this plant and documented it. To me, this joy and curiosity is what is driving human progress, not university education or not high school standard testing and not how many marks, multiple choice question you got guessed right. It's that emotion that is very, very important. It is our job as a scientist and artist and educator to capture that joy, right, and transform this into the driving force of learning. By the way, do you know what plant that is? No. We also don't know, right? We know that it's in Shingmun Reservoir. In order to go there, you have to hike for 1 hour. There's no public transport, or there's public transport, you have to walk for 1 hour. We have done DNA barcoding. It is a lot legal to collect anything from country park, is in the country park, but we broke the law, so she cut actually part of the plant, brought back to the lab. We have done DNA barcoding. We have narrowed down to a particular groups of plants. We still don't know, it's not documented to be moving. There are many hypotheses. For example, one hypothesis that you may think crazy, but we spent 3 months trying to test this hypothesis, and the hypothesis is there's a monkey hiding and moving it, right? You may laugh, right? But for the people who visited Shingmun Reservoir, Country Park, you know there are many monkeys, right? So we thought there is a super intelligent monkey. Every time she's hiding and then wait, right? And we investigated that there's no monkey. We use banana and other other methods to try to kind of attract the monkey. And nobody came out to claim the banana. We still don't know why it's moving. Our hypothesis is, in fact, it is a great subject for your DIY lab, right, to work on. We don't believe that plant can hear things and have a brain to respond to your greeting. We believe this plant is responding to vibration, air vibration. We believe that the trunk is made of material that is very, very tough to carry the weight, but extremely elastic. So if it is true, we want to characterize this material. Maybe that material can allow us to make better tools. That's the job of a scientist, an engineer, right? But the first person who discovered that is not a science student, not an engineering student, but an art student. So, see, I told you I love talking, right? So this, I always spend, so Maria, this is my 20 minutes. So, thank you so much for your patience. I don't want to claim all the credits. So we have a team of people who teaching this program. So there is a very multidisciplinary. We have a physicist, 2 artists. We also have a very good, wonderful teaching assistant. His name is Carsten. He's from Germany. Unfortunately, he's leaving. So if there's anyone who wants his job is to actually do all these things. Please email me. I don't know whether is it okay or yes? Okay.
So now will be an open dialogue between you guys and also Dr. Lam.
Yes.
If you have any questions or you want to share your own experience or your comments, yeah, you can ask.
The slime mold is just moving in a 2-dimension form.
Yeah.
Can it move?
Yeah, great question. What's the name?
Kelvin.
Kelvin. It's a great. We have done that. There's actually another student, and she. So basically, the slime mold project was done in the class of 2015. Semester A, which is from September to December. That's the semester of Occupy Central, right? All the young people were deeply affected, and one of the students asked exactly the same question, and she said she wants to, of course she's not interested in whether the slime mold can beat gravity and what's the force, and she's not interested in all that. She's interested in yellow and Occupy Central, and she said the collapse of Hong Kong values. And so when you have any collapse, it has to be 3-dimensional, right? So she built actually Agar, that is very ambitious. She wants to build a 3D model of the buildings in Central, and want the slime mold to climb on the top. What do you guess? Can they climb or?
I think so.
The answer is no. So they cannot climb up a vertical slopes. So in the end, she's not just a scientist, but also a very flexible person. So in the end, she put the slime mold on the top of the building. Then it climbed down. Anyone, any ideas? And also, I don't know whether I can answer on your behalf, or any things reagents, consumables, bacteria, slime mold, organisms, zebrafish that you've seen today, I will give them to her, to your lab. So if you want to play, you're not students anymore, but if you want to play and join the fun of doing stupid things with organisms, you're welcome, right? You can go to your lab and play. Yes.
So this is for first year students?
Most are first year students.
And what was kind of the selection criteria? So they were entering more of an arts with the...
Notice I'm going to answer, but it's okay. Do you want an honest answer or official?
Official and honest, right?
Official answer is we have a very, very stringent criteria. We only selected talented students who wanted to become. The honest answer is nobody choose this program. Nobody want to study this program. Whoever apply will get it.
One question. Just now you were talking about the frequency, right? I'm just wondering whether they're going to do any frequency on humans, the music and all that. And the reason why I'm saying is that I'm wondering whether the response with the human frequency and the environment frequency, which is basically now the fish, whether they actually, the frequency
That's very
so that they can actually coincide together and
Yeah, so-
synchronicity instead of like which all these environmental issues
yeah.
where we are actually out sync. So I'm
That's-
just curious.
There's a DIY project. We haven't done that yet. I strongly suggest Nero to do that. If you go to his YouTube channel, Nero Low, you can see his more than one videos. And one of the things, as I said to you many times before, one of the things I impressed about this student is we all sink into the trap of trying to control nature. When a fish follow your movement, we say, oh yeah. But Nero is the only student in the class who said, why must we control the fish? So why can't we have the fish swimming in harmony with the human music? And the fact that he, instead of playing the music to the fish, but instead of doing that, but transform the music into light and pattern, is because fish cannot hear, so they sense vibration through their skin, but they haven't evolved an ear to detect air vibrations, right? I'm going to tell you also a crazy project by the students, another group of students who are so crazy that I don't include. That is about if you play music to plants, can you detect any difference in their growth?
It's not crazy. I think.
You think, right?
Yes.
So when you
Experiment it, right?
so they did it.
Yes.
They did it, and of course I'm scientist and need to be responsible for. So they have done it once. 3 plants with 3 music, well, 2 kinds of music and silence. But then to make scientific conclusion, you need to do it 100 times, and
Yes.
you need to
Data.
you need the data. If the data is consistent,
Yes.
80 times out of 100, then you make a claim, right? Did it one time. So to me, it's more like an art experiment, an art project. Then, but anyway, silence. One is heavy metal rock,
Right.
and one is classical music. So can you guess, is there any difference?
How long do you play it for?
For they have seedling, I think play for 3 days,
Non-stop.
non-stop. Constant playing for 3 days non-stop, and they took great care in
soil
making sure the soil, water, and light temperature. They even have a temperature probe to record the temperature change over the day of the 3 occasions, and they have soundproof boxes that is light transparent. So the answer is, can you guess? There's one piece of music that have a slight kind of effect on growth, make the classical.
There should be some other experiment on water for the same purpose. So, but the so the water also shows difference.
Yeah, but but once again, there's no replications, and we don't know. For us, we encourage the student to do that, not to kind of tell the world that playing Mozart and it's better for the plants. There's a pseudoscience, right? I was trained to discredit those claims. But the journey of how the consideration they need to make, standardizing temperature and water, humidity, in order to single out differences between music, is a good kind of thinking process for the students. So I think it's still a valuable exercise, but I would take the result with a huge amount of salt. Yes? Maybe this gentleman first.
So you were saying this is a very new course, possibly the first, but in a way it's kind of also a really old thing. It's going back to the times of Leonardo da Vinci,
Yes.
and
Yes.
the professionalization of science is a very new thing.
Yes.
And it's it was all curiosity driven, and it's getting more and
Yes.
more focused, and even more so. They're not even doing basic science anymore. All the funders and everybody are
This-
doing more.
exactly.
Everything applied. So what can we do to bring creativity back into science?
Bringing art into STEM education. What you said is a really great summary of the challenge we are facing now. In fact, I prepared. So I'm going to show you. If you're a scientist, we learn. So, as you said, 200 years ago, when we didn't know so much about the world, scientists are, in fact, at the same status as a philosopher. Yeah. We think about nature, we ask important questions, and we devote our life to those questions. But now, science, there seems to be a lot of progress over the past 200 years. But now, science education is nothing to do with curiosity, but to do with learning of techniques. And to me, there are fundamental differences. So it's a screen capture of a big company. This big company is called Thermo Fisher, that used to be called Life Technology, that used to be called Invitrogen. So for biologists, you know about this. The problem, I think, is this is the biggest supplier of equipment and consumables for biomedical research in the world. So it's like a supermarket. So now, if you want to do a PhD, or if you want to ask a biomedical question, you simply have a question, and then you have a ready-made tools, kits, to answer those questions. I want to know what are the difference in gene expression between this cancer and that cancer. Immediately, I will click on that box, and I will click, and then I will buy a kit, and the kit will contain a box. Open the box with solution, label PQR, and then there's a booklet, and then you just mix everything together, and then you make the gene. You basically discover genes that are expressed in these cells and not that cells. There's no thinking about it. It's about following procedures and buying the right kits. These companies supplied the same set of kits. Of course, the kits contains the inventories of the inventory contains thousands of kits. There's a kit for every technique. There's a technique for every question. So now, if you do a PhD, or if you're a professional scientist like me, you're basically shopping and then doing the experiment. The experiment was the kits are so well made that if you didn't make crazy, stupid mistakes, you get the result you want, right? So very fast. If you want to clone a gene, in the past it take one PhD 4 years in order to clone a gene. Now it take 1 week, right? It's a major steps. But this improvement is in terms of kit manufacturing, is an engineering, is a manufacturing, is a victory of manufacturing. So basically, because we are all using the same set of kit, we have an illusion of progress, because every experiment we save so much time, but our thinking is limited by the availability of kits. Whilst artists, or you guys, you don't think in terms of kits. you think in terms of a maker's culture. One thing that I learned a lot from artists is when an artist have a question they want to solve, they always make their own tools, right? For example, Bobo, she made the map of C.Y. Leung's home. She didn't buy a kit, right? And she's not stopped because the kit is not available. She's going to try she's going to try all her best to make her project come true, including stopping people at her home to go to toilets, right? So everything is homemade. It gives you an illusion of not making very good progress. But because everything is original, everything is tailor-made for her particular experiment, in a way, her kind of ambition is much higher than those of professional scientists. I'll give you one example. When you do cell culture only for the people who are biologically trained, cell culture, basically putting cells in a petri dish, right? A single petri dish. Sometimes you have multiple culture and you have multi-wells, right? You have one petri dish divided into wells. So there are 6 wells, a dish with 6 wells. There are dish with 12 or 24 or 96 wells, right? So that you have 96 slots of doing parallel experiment. So, because that's the standard, that's the industrial standard. However, if there's an experiment whose design required you to have 97 samples, I can't do it because I will do 96 well plus one well. But if I do the incubation at the same time, it doesn't work, right? It's a different kind of conditions. So I can never do a parallel measurement of cell behavior of 97 samples or 25 samples or 13 samples. It's crazy, right? My creativity, my design is limited by the industrial standard. But those artists, they will never kind of shop for petri dish, right? If it doesn't have enough well, she's going to 3D print a dish that contains 97 wells with a bigger well just for the sake of it. Or she's going to print a petri dish with a tripod just because it suits them, right? So these kind of things, the kind of flexibility and the kind of maker's identity is something a scientist in the 21st century must learn from the artists. Otherwise, we can't survive, actually. You have the gentleman behind.
I remember probably 20 years ago, there was this artist called Eduardo Cat.
Sorry,
Eduardo Cat.
Yeah, the GFP rabbit and
Yes, so almost 20 years ago, he was doing experiments with transgenic animals. Actually, he created this bioluminescent rabbit. So the potentiality of biology and art are crazy also. It's going to be like programming instead of a software. It will be also applied to that area and scale. I think even the project with the fish being triggered by the pattern of the iPhone can raise a few moral questions.
Exactly, yes.
I mean, I'm not taking any position. It's not talking about moral is not even a moral issue. It's more a philosophic position and interpretation of things. So I'm asking you personally, where would you put the line of the sand, the limit, where you think something is worth doing for the sake of art, not even science, for the sake of art, and when it's very difficult to
It's a great question. What's your name?
Luca.
Luca, it's a great question. Thank you very much. In fact, Mariana will probably remember we have this discussion in our lecture as well. In fact, one particular student did the following project, but I don't know. I don't want to bore you guys. Do you want a short answer or long answer? Short answer, right? Short answer. Short answer is I don't know. I'm going to tell you a little bit more about the fish experiment to illustrate. But what I'm going to say will disgust you. Is it okay? You're going to hate me, not just me, but also all the scientists in the world. So experimentation on living organisms is actually restricted by an ethical committee. We have a university ethical committee, and we in Hong Kong in general there is a committee under the Department of Health of the Hong Kong government. For example, if you want to kill a fish, just take a fish and then you're gonna kill it very humanely in order to study, for example, the anatomy of the fish and maybe the biochemical composition of the brain, for example. You don't need any license, right? Just because it's an animal, we slaughter animal for food anyway. So instead of eating, we dissect. It doesn't matter. So no. However, if you take an organism and do experiment on it, for example, if you take a mouse and you inject a toxic compound every day until it dies, right? Then you better answer to the committee, right? You're going to kill the animal who may suffer from pain for nothing, right? Then if it is for fun, for art project, no. But if you're going to assess this toxin as a potential drug that will eventually benefit a lot of human beings, then the ethical committee will balance the benefits and the pain suffered by that particular mouse, and then they will decide may or may not give you the license, right? So, however, the fish, so what's the position? That's the part that you're going to hate me about. According to the regulation, if a fish that is less than 4 days old is not considered to be a living thing. Therefore, if the fish or any vertebrate organisms that is less than 4 days old, they are considered as a dish of cells. So the fish, for example, the small fish that is swimming after we have done the experiment, we flush them down to the toilet. Well, we don't like it, but we are not breaking any laws. And the reason behind is simply because of the reproduction system of fish. Human beings, we have to fall in love and we have to get married and then to give a baby, right? Or may not fall in love, but anyway, you have to do very, very complicated cultural and legal process to make a baby. But fish, they don't do that. The female fish release thousands of eggs every day, and the male fish release millions of sperm every day, and the fertilization is outside the bodies, right? The parents will swim away, and the fertilization will outside in the water outside the bodies. 99.9% of the eggs and sperms, and also the fertilized eggs, are eaten by other fish, right? So because of the external fertilization, there's such a wastage to the process that this kind of one fish that we play with and flush down the toilet is statistically negligible compared to the wastage of the reproductive process. By the time it reached 4 days, day four, the small fish will develop, they continue to grow, right? There's something happen in day 4. In day 4, they will grow a mouth and they will start to eat, right? Before day 4, they cannot eat. There's no mouth. They're still using the egg yolk that is actually from the mother's egg for its survival. By the time it starts to have a mouth, it can eat, then it is independent life form, right? Because if you kill somebody who can eat, then you destroy a life, right? So that's the kind of a lot of, if you go to a lot of cosmetic company, they say no animal testing, right? No animal testing. But how can you sell a chemical where people put on their skin every day with no toxicity testing, right? So they make something you put on the skin every day, but they never tested the potential toxic effect of this compound on anything. Can you trust it? But of course, we hate animal testing so much that we believe that. In fact, they said no animal testing because they test on fish. They test on fish that are younger than 4 days old because they're not considered animals, right? So therefore, the legal aspect is for this fish is fine. We play around this and we laugh, put it on Facebook and YouTube and flush down to the toilet, right? So we don't enjoy it, but we're not breaking any rules. One of the major discussion we have in the class, and once again, it's unique for an art class because in science class we don't care, right? The students are so busy with their homework and with the gene sequences, and in that particular lecture we play with silverfish. We discuss, and interestingly, the student in that particular class were actually asking the following question: when we are hurting the fish, we are breaking some rules or breaching some potential ethical issues. But one student said there are actually many artists, performing artists, they are using their bodies to perform, especially on the subject of pain and endurance, right? There are people, I think some students said there's an art performance in the early 1970s where a female artist stripped naked and used a knife to cut her belly, right? So, and then in science, scientists are not allowed to experiment on ourselves. So if we experiment on ourselves, we can't publish the papers. We have to experiment on other people, right? So anyway, so there's actually a very, very different concept between scientists and artists when we collaborate with other organisms. And she actually wrote an essay about it, and that's the long answer. I'm sorry, I forgot. But once again, I think the use of art, the importance of art is not to make beautiful things and it's not a show, right? In fact, all good art are difficult art. They ask difficult questions and make comments and ask questions about what we are doing in everyday lives that in an unexpected way and stimulate people to think